Hello, my name is Joshua mUGABI. I welcome you to Topic 1, Cell Biology. Our learning outcome: analyze the properties and the functions of chemical compounds, water, lipids, proteins, including enzymes from mammals, in a cell, focusing on their roles in maintaining cellular structure and metabolic processes in living organisms. We are looking at learning activity A. In groups, learners search for and discuss A. Interaction of water with polar and nonpolar substances. Simple and conjugated lipids, their properties and functions. Structural categories of proteins and their properties and functions. Create audiovisual presentations and share them with other groups. We shall start with interaction of water with polar and nonpolar substances. Item 1. During a school laboratory practical, students attempt to mix salt, sugar, and cooking oil separately with water. Salt and sugar dissolve completely, but the oil forms droplets and floats on the surface. A learner asks why living cells are mostly made of water if many substances in organism do not mix in it. Task: classify salt, sugar, and cooking oil as polar or nonpolar substances. Explain why water dissolves some substances but not others. Describe how this property of water is important in living organisms. Predict what would happen if detergent is added to oil-water mixture and explain why. Let's start with classify salt, sugar, and cooking oil as polar or nonpolar substances. Salt and sugar are polar substances. Cooking oil is nonpolar. Explain why water dissolves some substances but not others. Water is a polar molecule and forms hydrogen bonds with the polar substances, allowing them to dissolve. Nonpolar substances do not form hydrogen bonds with water and are therefore insoluble. Describe how this property of water is important in living organisms. Water dissolves nutrients such as glucose and salts for transport in blood and cytoplasm. Nonpolar substances like lipids form membranes and energy stores. Predict what would happen if detergent is added to the oil-water mixture and explain why. Detergent will cause the oil to disperse into tiny droplets. Detergent molecules are amphipathic with both polar and nonpolar regions, allowing them to interact with both water and oil. Now let us look at simple and conjugated lipids. We shall look at the properties and the functions. Item 2. A farmer note says that animals raised in cold areas have thick layers of fat, while laboratory analysis of cell membranes show high amount of phospholipids, glycolipids. Students are asked to explain why different lipids are used for different purposes in organisms. Task: distinguish between simple lipids and conjugated lipids. Identify the properties of each lipid group. Explain the functions of triglycerides, phospholipids, glycolipids. Justify why phospholipids are suitable for forming cell membranes. Let's start with distinguish between simple lipids and conjugated lipids. Simple lipids are made of fatty acids and alcohol only. For example, triglycerides, waxes. Conjugated lipids, these contain additional groups such as phosphates or carbohydrates. For example, phospholipids or glycolipids. Identify the properties of each lipid group. Simple lipids are hydrophobic, meaning insoluble in water, and energy rich. Conjugated lipids are amphipathic, having both hydrophilic and hydrophobic regions. Explain the functions of triglycerides, phospholipids, and glycolipids. Triglycerides, they are long-term energy storage. They are used for insulation and protection. Phospholipids, they are for the formation of cell membranes. Glycolipids, these are for cell recognition and communication. Justify why phospholipids are suitable for forming cell membranes. Phospholipids, have hydrophilic heads and hydrophobic tails, allowing them to form a bilayer that separates the cell from its environment. Let's now go to structure categories of proteins, their properties and functions. Item 3. A patient with a genetic disorder produces hemoglobin that cannot efficiently transport oxygen. Laboratory analysis shows a small change in the amino acid sequence of the protein. Students are asked to explain how changes in protein structure affect function. Task: list the 4 levels of protein structure. Describe the properties of each structure level. Explain how a change in the primary structure can affect protein function. Give one example of a protein and relate its structure to its function. List the 4 levels of protein structure. We have the primary protein, secondary proteins, tertiary protein, and quaternary structures. Describe the properties of each structure level. Primary structure: sequence of amino acids linked by peptide bonds. Secondary structure: alpha helices and beta sheets stabilized by hydrogen bonds. Tertiary structure: overall 3D folding stabilized by various bonds. Quaternary structure: association of 2 or more polypeptide chains. Explain how a change in the primary structure can affect protein function. A change in the amino acid sequence alters folding, changing the protein's shape. This affects active sites or binding regions, reducing or stopping functions. Give one example of a protein and relate its structure to its function. Hemoglobin has a quaternary structure, allows binding of oxygen. Any structure alteration reduces oxygen transport efficiency. Overall learning outcome. Water's polarity determines solubility, and cellular organization. Lipids differ in structure, leading to different biological roles. Protein structure determines function, and small changes can have major biological effects. Thank you for participating in Topic 1 Cell Biology. We have been looking at learning outcome A. Analyze the properties and the functions of chemical compounds, water, lipids, proteins, including enzymes from mammals, in a cell, focusing on their roles in maintaining cellular structure and metabolic processes in the living organism. We have been looking at learning activity A. In group, learners search for and discuss interaction of water with the polar and non-polar substances, simple and conjugated lipids, their properties and function, structure categories of proteins, their properties and functions. You're supposed to create an audiovisual presentations and share them with other groups. If this has been useful, please visit me at mUGABI Joshua Biology on YouTube.